Every time you sprint, every time a yeast cell fragments sugar into alcohol, every time a red blood cell which has no mitochondria at all keeps itself alive, the same 10 reactions are running. They are so ancient and so universal that they happen in almost every living cell on earth, from bacteria to your own neurons. This is glycolysis. The splitting of glucose. It takes only 1,6 carbon sugar and through 10 precisely controlled steps breaks it into 3 carbon molecules of pyruvate, harvesting energy along the way. But here's the thing most students get wrong. They memorize the 10 names and forget the chemistry. In this video, we'll do it properly. We'll go through all 10 steps, and at every step, we'll look at the actual structure, which carbon is being attacked, where the phosphate goes, how the molecules is rearranged, so that by the end, the pathway isn't a list you memorized. It's a story you understand. So let's begin. Before the steps, let's frame the whole thing. Where does it happen? Glycolysis takes place entirely in the cytoplasm or the cytosol of the cell, not in the mitochondria. That matters. It means glycolysis can run without oxygen, which is why it works in your muscles during a sprint and in organisms that never see oxygen at all. What's the overall transformation? One molecule of glucose of 6 carbons splits into 2 molecules of pyruvate, 3 carbon each. That is, carbon in, carbon out. Nothing is lost. The 6 carbons are simply split into 2 threes. The pathway has 2 phases, and you must hold this division in your head because it explains the entire energy story. Phase 1. The preparatory or investment phase. From step 1 to step 5. Here the cell spends energy. It invests 2 ATP to energize and destabilize the glucose. Then split it into 2 3 carbon sugars. You have to spend money to make money. The second phase is payoff phase. Here the cell harvests energy. It generates 4 ATP and 2 NADH. And crucially, because glucose was split in 2 at the end of phase 1, every reaction in phase 2 happens twice. Once for each 3 carbon fragments. Keep that into 2 in your mind. This is why you'll see 2 written in bracket besides these structures in the notes. So the net yield, and remember this number, is 2 ATP and 2 NADH per glucose. We'll build up to that figure step by step. One more concept before we start. 3 of the 10 steps are irreversible. Step 1, Step 3 and Step 10. These are the regulatory checkpoints, the one-way valve that controls the whole pathway. Watch for them. Now let's talk about the structures and the phases. Phase 1. The preparatory or investment phase. From step 1 to step 5. Step 1. From glucose to glucose 6-phosphate. The structure we start with is glucose. Look at the ring. Glucose is a 6 carbon aldohexose drawn here in its cyclic pyranose form. A 6-membered ring. Number the carbons. The key one for this step is carbon 6, sitting up top as the CH2OH group. The free primary hydroxyl hanging off the ring, that CH2OH is our target. The enzyme Hexokinase transfers the terminal phosphate group from ATP onto that C6 hydroxyl. ATP becomes ADP and a phosphate is now covalently bonded to carbon 6. The structure we produce, glucose 6-phosphate. Notice what's changed. That CH2OH at carbon 6 is now CH2O PO3 2-. A phosphate group is struck onto that carbon 6. That's the entire structural change. One hydroxyl converted to a phosphate ester. Why do this? There are 2 reasons and both are very important. The phosphate is charged and negative. So glucose 6-phosphate cannot leak back onto across the cell membrane. Phosphorylation traps glucose inside the cell. And second is, it raises the energy of the molecule, priming it for what's coming. 2 things for your exams to remember. These steps require magnesium as cofactor. Magnesium stabilizes the negatively charged ATP, our first regulatory checkpoint. Investment so far is 1 ATP. Step 2. Glucose 6-phosphate to fructose 6-phosphate. The structural change here is a rearrangement, an isomerization. Watch the 2 structures side by side. Glucose 6-phosphate is an aldose. Its reactive carbonyl C-O is an aldehyde at carbon 1 and it sits in a 6-membered pyranose ring. We are going to convert it into a ketose. The enzyme phosphohexose isomerase, also called as phosphoglucose isomerase, shifts the carbonyl group. It moves the C-O from carbon 1 to carbon 2. The structure we produce, fructose 6-phosphate. Now look, the ring has become a 5-membered furanose ring and the carbonyl is a ketone at carbon 2 instead of an aldehyde at carbon 1. The phosphate is still on carbon number 6, untouched. Aldose has become ketose. Why bother? This is preparatory move. By shifting the carbonyl to carbon 2, we free up carbon 1 and give it a hydroxyl group. And in very next step, the carbon 1 hydroxyl is going to receive a phosphate. You can't phosphorylate carbon 1 while it's locked in an aldehyde. The isomerization steps up the next reaction. This step is reversible. No energy spent, just a rearrangement. Step 3. Fructose 6-phosphate to fructose 1,6-bisphosphate. The structure going in. Fructose 6-phosphate. Focus on carbon 1. That free CH2OH group we just liberated in the last step, that is our target now. The reaction, and this is the most important regulatory step in all of the glycolysis. The enzyme phosphofructokinase 1 or PFK1 transfers a second phosphate from ATP onto the carbon 1 hydroxyl. ATP becomes ADP again. The structure we produce is fructose 1,6-bisphosphate. Now the molecule has a phosphate on both ends, one on carbon 1 and one on carbon number 6. That's why it's called bisphosphate, two phosphates at two separate positions. The sugar is now symmetrically charged at both ends. Why this is the key step? This is the committed step. Once fructose 1,6-bisphosphate is made, the molecule is locked into glycolysis. There is no other use of it. Glucose 6-phosphate could have gone off to make glycogen or enter the other pathways. But fructose 1,6-bisphosphate has only one destination. It's also the rate limiting step, the slowest controlling reaction, and PFK1 is the master regulatory enzyme, switched on when the cells need energy and switched off when energy is plentiful. This step is irreversible. Checkpoint number 2. And it cost our second ATP. So our total investment now is 2 ATP. The spend is done. From here on, everything either splits off or pays off. Step 4. Fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. This is the lysis step, the actual splitting of the sugar. The 6-carbon molecule is cut in half into two 3-carbon molecules. Let's look at the structure. Fructose 1,6-bisphosphate opened into its chain form is a 6-carbon backbone with phosphate at each end. The enzyme cuts the bond between carbon 3 and carbon 4, snapping the 6-carbon chain into two 3-carbon pieces. The enzyme Aldolase performs this cleavage. An aldol cleavage, which is where its name comes from, the two structures we produce and they are not identical. Glyceraldehyde 3-phosphate or G3P. Carbon number 4, 5 and 6 of its original sugar. Its structure POCH2 CHOH CaHO. Note the key feature. It ends in an aldehyde group which is CHO. This is aldose triose and critically this is the molecule that continues down the pathway. Dihydroxyacetone phosphate or DHAP. Carbon 1, 2 and 3 is structure POCH2 C-O CH2OH. Note the difference. Its carbonyl is a ketose C-O in the middle. This is ketose triose. So we have two different 3-carbon phosphates. One an aldehyde, one a ketone. Only G3P can proceed. But the cell isn't going to waste half of its glucose, which is exactly what step 5 fixes. Step 5. Dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. The structure change. another isomerization. Just like step 2, we take the DHAP with its central ketone and rearrange it into G3P with its terminal aldehyde. The enzyme triose phosphate isomerase interconverts the two triose. It converts the dead end DHAP into another molecule of G3P, the form that can continue. This is the pivotal bookkeeping moment because DHAP is converted to G3P. We now have two molecules of glyceraldehyde 3-phosphate or G3P heading into phase 2. This is precisely why every structure in the second half of my notes has 2 written in brackets. From this point on, every step happens twice, once for each G3P. This is the end of phase 1. Let's take stock. We spent 2 ATP and we have turned 1 glucose into 2 molecule of G3P. No energy harvested yet, but the molecule is now primed and split and ready for the payoff. Step 6. Glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. The structure going in glyceraldehyde 3-phosphate POCH2 CHOH CHO. Focus on the aldehyde group CHO at carbon number 1. Two things are about to happen to it simultaneously. It will oxidize and it will phosphorylate. This is the only oxidation step in glycolysis and it's where NADH is made. The enzyme glyceraldehyde 3-phosphate dehydrogenase or GAPDH does two things at once. It oxidizes the aldehyde group. The electrons and a hydrogen removed in this oxidation are transferred to NAD+, reducing it to NADH plus H+. Because we have 2 G3P molecule, 2 NAD+ become 2 NADH plus 2H+. It simultaneously attracts an inorganic phosphate from the cytoplasm, importantly not from ATP, onto the newly oxidized carbon. The structure we produce is 1,3-bisphosphoglycerate POCH2 CHOH C double bond O OP. Look carefully at carbon number 1. The old aldehyde is now a highly energy acryl phosphate, a carbonyl carbon bonded to phosphate through an oxygen. There are now phosphates at position 1 and position 3, hence 1,3-bisphosphoglycerate. Here's the elegance. The energy released by oxidation, the aldehyde wasn't lost as heat. It was captured in the new high energy acryl phosphate bond. That stores energy is what the cell will cache it for ATP in very next step. Note also, no ATP was spent here. The phosphate come free from the cytoplasm and we have banked our 2-NADH. Step 7. 1,3-bisphosphoglycerate to 3-phosphoglycerate. The structure going in is 1,3-bisphosphoglycerate, carrying that high energy acryl phosphate on carbon number 1 that we just built. The enzyme phosphoglycerate kinase transfers that high energy phosphate from carbon number 1 directly onto the ATP, making ATP. Because there are 2 molecules, 2 ADP becomes 2 ATP. This is worth naming precisely because it's a favorite exam term. This is substrate level phosphorylation. ATP generated by transferring of phosphate directly from a high energy substrate to ADP, as opposed to the oxidized phosphorylation that happens later in the mitochondria. It needs no oxygen, which again is why glycolysis can power anaerobic cells. The structures we produce is 3-phosphoglycerate POCH2 CHOH COO-. The high energy phosphate on carbon 1 is gone. It's now in ATP. Carbon 1 is now simple carboxylate group COO-. Only the phosphate on carbon 3 remains. Step 8. 3-phosphoglycerate to 2-phosphoglycerate. The structure change. The phosphate simply relocates. We move the phosphate group from one carbon to a neighboring one. The enzyme phosphoglycerate mutase shifts the phosphate group from carbon 3 to carbon 2. A mutase is an enzyme that relocates a functional group within a molecule. The structure we produce is 2-phosphoglycerate CH2OH CHOP COO-. Compare it to the 3-phosphoglycerate we started it with. The carboxylate at the end is uncharged, but the phosphate has hopped from carbon number 3 to carbon number 2. The carbon 3 is now plain hydroxyl. Carbon 2 now carries the phosphate. But why? This looks like a trivial move, but it's strategic positioning. By placing the phosphate on carbon 2 right next to carbon 1, we set up the next reaction, a dehydration that will turn into a high energy compound. These steps require magnesium and it is reversible. Step 9. 2-phosphoglycerate to phosphoenol pyruvate. The structural change, a dehydration. We remove a molecule of water, and in doing so, we create one of the highest energy phosphate compound in all of the biology. The enzyme Enolase removes a molecule of water from 2-phosphoglycerate. This dehydration redistributes the energy within the molecule, forming a high energy enol phosphate. The structure we produce is phosphoenol pyruvate or PEP, CH2 double bond C, OP, COO minus. Look at what the loss of water creates, a double bond carbon between carbon 2 and carbon 3, with the phosphate now sitting on the enol carbon. That combination, a phosphate attached to a carbon that's part of C double bond C and enol phosphate, is extraordinarily energy rich. Phosphoenol pyruvates hold its phosphate with more energy than ATP does. That's exactly why in the final step it can force its phosphate onto ADP so readily. The dehydration by enolase has loaded the spring. Step 10. Phosphoenol pyruvate to pyruvate. The structure going in is phosphoenol pyruvate, carrying that ultra high energy phosphate we just built. The enzyme pyruvate kinase transfers the high energy phosphate from PEP directly onto the ADP, making it ATP. Two molecules again. So 2 ADP becomes 2 ATP. This is the second substrate level phosphorylation of the pathway. The structure we produce is pyruvate, CH3 C double bond O COO minus. This is the end product, a 3-carbon molecule with a methyl group, a central ketone and a carboxylate. The phosphate is gone into ATP and enol has rearranged into a stable keto form. Since we had 2 molecules traveling through, we finished with 2 pyruvate. This step is irreversible, our third and final regulatory checkpoint, and it's the second big control point that commits the pathway to completion. Glycolysis is complete. 1 glucose has become 2 pyruvates. You might ask what happens to pyruvate. Where does it go? It stands at a metabolic crossroads determined by oxygen. With oxygen, that is aerobic, pyruvates enter the mitochondrion, is converted to acetyl coenzyme A and feeds the Krebs cycle and electron transport chain, releasing far more ATP. The second is without ATP, in muscle, anaerobic. Pyruvate is reduced to lactate, forming lactic acid fermentation. This regenerates NAD+, so glycolysis can keep running when oxygen is short, for example during a hard sprint. And without oxygen in yeast, pyruvate becomes ethanol plus carbon dioxide, that is alcoholic fermentation, the basis of brewing and baking. Let's quickly recap. Phase 1, investment phase. Spend 2 ATP, splits the sugar. First step, glucose to glucose 6-phosphate by hexokinase. Second step, glucose 6-phosphate to fructose 6-phosphate by phosphohexoisomerase. Third step, fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1. 4. Fructose 1,6-bisphosphate to G3P plus DHAP by Aldolase. Step 5. DHAP to G3P. Second phase, payoff phase. Everything is into 2. Harvested 4 ATP plus 2 NADH. Step 6. G3P is converted to 1,3-bisphosphoglycerate by GAPDH. Step 7. 1,3-bisphosphoglycerate to 3-phosphoglycerate by phosphoglycerate kinase. Step 8. 3-phosphoglycerate to 2-phosphoglycerate by phosphoglycerate mutase. Step 9. 2-phosphoglycerate to phosphoenol pyruvate by Enolase. Step 10. Phosphoenol pyruvate to pyruvate by pyruvate kinase. Net outcome per glucose is 2 ATP, 2 NADH and 2 pyruvate. If this chemistry of glycolysis finally makes sense now, not just the names but why each structure changes the way it does. That's exactly what our channel is for. Practice by drawing the pathway from memory, structure by structure, and that's how it sticks in your mind for the exam. Keep learning, keep achieving. I'll see you in the next video. Thank you.